Hello, my lovely anatomists and physiologists. Welcome to our video on capillaries. When we talk about capillaries, we've said this is the location for exchange. We can say a new word here. So we can say like the job of the capillary is to ensure good perfusion, where perfusion is talking about adequate blood flow to our tissues. Adequate blood flow to our tissues. And we said there's three types. There's continuous, fenestrated, and sinusoid. So what we're going to see is the continuous is the least permeable, and the sinusoid is the most permeable. And so we're on this continuum from least to most, and the fenestrated then can fit like in between. The fenestrated can fit like in between. Then when we talk about the capillary, we said that there's a basement membrane, which we've already said is a permeable connective tissue layer, meaning material can move through it. And then we have the endothelial cell, which remember is our simple squamous. That means a single layer of cells, one layer. And then we see occasional muscle. Muscle fibers. We're talking about little muscle cells here. On occasion, right? And so we're talking about an extremely small vessel. We've also said it has a lumen of like 4 to 10 micrometers in diameter. It's very, very small. Remember, that's even smaller than our red blood cell diameter, which is like 8 micrometers. When we're looking at our capillaries, there's 3 types. I've organized this into a table so I can do my quick compare and contrast. And so I have 3 columns: continuous, fenestrated, and sinusoid. Of course, you can organize your notes the way that you best like. When we look at the continuous capillary, we're going to see that it's the most common. And we're going to see that it's a complete endothelial lining. And it has tight junctions. Tight junctions are like these water sealed connections between cells, so that there shouldn't be a lot of leakage that's happening between cells. Although we will see that this is incomplete. So even though we have like these tight junctions, which are these watertight seals, it's incomplete. And we're going to talk about those incomplete areas as intercellular clefts. And I think here we should think about like that synaptic cleft, right? Meaning just like that tiny space between the synaptic knob and the postsynaptic cell. So in the case of the intercellular cleft, let's just think, oh, it's a tiny space between cells. If I'm drawing my capillary, if I'm looking at it like looking at the top of a straw, I'm going to maybe draw just 2 simple squamous epithelial cells, or my endothelial cells connected. And so this space in the middle, that would be my lumen, right? So let me label that lumen. Really tiny. And so as we're talking about exchange happening at the continuous capillary, when we talk about exchange happening at the continuous capillary, we're thinking about material that has to pass between the cells. or through the cell membrane. So it's going to pass through the cell membrane of the endothelial cell, past that basement membrane, which is permeable, and then out to your tissues. And so what we can see that passes is, of course, your gases. So oxygen gas can cross, it's lipid soluble. Carbon dioxide gas can cross because it's lipid soluble. We see that water can pass through because it is small, We see that your hydrophobic or lipid soluble hormones can pass. And then we see things like glucose can pass. So we're going to have like a carrier protein in the cell membrane of these cells to allow for that glucose to move. And we also will see a lot of vesicular transport in these kind of capillaries. So vesicular transport, remember, involves endocytosis and exocytosis. And so that's where you have quite a lot of material packaged up in a vesicle that fuses with the membrane and either enters the cell or exits the cell, depending, right? So we'll see quite a lot of that happening here as well. When we move to the fenestrated capillary, everything that we talked about with the continuous is true, but what we'll also see is that they have Finstra, which I misspelled here, which is really talking about pores. I think technically this is like an Italian word or derived from an Italian word for window. So maybe think about in the days before having a window pane with glass, or maybe a window that opens, right? So this is like a hole in the vessel. And so this is going to obviously increase permeability. So now your large molecules can more easily cross, and that movement can happen faster. So everything like carbon dioxide gas, oxygen gas, water, hydrophobic hormones, glucose is still crossing, but now even bigger stuff can cross. And so we'll see this type of capillary strategically located in places where you want to have a lot of movement. So for instance, the small intestine, which I'm abbreviating here as SI, this is your location for two things, like you finish digestion and you absorb nutrients. And so we get a lot of hormones from our blood to help coordinate digestion. So a lot of stuff has to leave the blood and go into the small intestine. But then the reverse of that, we're absorbing nutrients. So then nutrients from the small intestine have to move into the bloodstream. The kidney is a huge place for movement. We're producing urine at the kidney, right? We're going to see choroid plexus, which is located, remember, in your brain ventricles. This is where your cerebrospinal fluid is being produced, right? We're going to see this in a lot of our endocrine glands. So hypothalamus, we're moving hormones into the bloodstream. This should be making perfect sense. Pituitary gland, pineal gland, and thyroid gland. Okay. Now, if I drew this one, I'm going to draw the same kind of picture. So I'm going to make it just a little bit bigger so we can see the effect. So I'm going to do my two endothelial cells, my two simple squamous cells. That's my whole tube. That's my whole capillary. But now I'm going to take my eraser and I'm going to make openings. I'm going to make my Fenestra. Okay. And so now we get the cell regulating based on the cell membrane, but we also have these openings at the cellular level. Those are the fenestra. I'm gonna change my window that I drew up here. I'll make it just like be an opening, an open window with curtain. Okay, all right. Last is the sinusoid, and we said most permeable here, most permeable, and so also least common. We want to be super selective about where we make this. These are going to be flat. These are going to have extensive intercellular gaps. So now we're not even talking about gap in a cell membrane, but gap between cells. We have fenestra here. So there's still those pores, right? We have an incomplete basement membrane. Okay. And here, these openings are so big, we get the movement of plasma proteins. Hey, y'all, pay attention. Pay attention. This is the first time. This is the first time we can see plasma proteins moving in or out of the blood. So the plasma proteins cannot move through continuous. Plasma proteins cannot move through fenestrated. Remember, they're important in regulating osmolarity. So we're going to pick up that detail and make sense of it in a little bit. And we'll also see movement of the plasma proteins as well as, let's say, formed elements. You could say cells here, but what we're really talking about in this case are red blood cells. And so we're going to see these strategically located at the liver. That's where we make those plasma proteins. So they have to have an avenue to get into the bloodstream. We're going to see it at the spleen. That's where we're going to pull out all those old damaged, worn out red blood cells, right? We're going to see it in the bone marrow because, hey, that's where we're making those red blood cells and they have to enter. And we'll see them in lymph nodes as well. And so our book describes it as Swiss cheese. Oh, I love this example. And so I'm not confident in my skills of doing any kind of drawing here. So what I want you to do is use your imagination. So imagine a piece of Swiss cheese, like a sandwich slice, and then roll it in. Oh, poke some extra little holes in it, like with a toothpick, and then roll it into a tube. And so those little toothpick holes, that would be like the fenestra. And then like the big holes that's in the cheese, that's like those extensive intercellular gaps. So clearly, very, very permeable. And so clearly, this should be super, super regulated. I am going to highlight this movement of plasma proteins. So if you're not picking up on it, like this is a super important detail. So please, please, please make sure you're making a note of that. All right, up next, let's look at the capillary bed itself. And as always, take care of yourself and each other.